Timo Discheit es el responsable de Big Data Analytics en el Instituto de Neurociencia y Medicina de Jülich, en Múnich, en Alemania. Él tiene un doctorado en visión computacional por la Universidad de Bonn. Es el responsable del grupo de investigación de imagen biomédica, análisis de neuroinformático de imágenes cerebrales y reconstrucción de imágenes 3D del cerebro. Y bueno, lidera también el servicio, uno de los servicios importantes de eBrains, el Service Category número 2, que incluye pues todo lo que es relacionado con el desarrollo de Atlas y los correspondientes interface para explorar e integrar datos y analizar datos cerebrales. Él es compañero nuestro de HP, lleva bastante tiempo en el proyecto y bueno, para nosotros es un placer y un honor tenerte aquí. La presentación evidentemente va a ser en inglés. So, Timo, excuse me for introducing you in Spanish. I think it's better for us in this moment, but as you know, you can present everything from now onwards in English. Don't speak very much in German, please. Otherwise, we will have some problems to understand you properly. Thank you very much. The floor is yours. And well, welcome to our great Spanish innovation community in HBP. They are all experts in HBP things after a long time of interaction with this outstanding group. So yeah, the floor is yours and thank you. And we are looking forward to know more about the Atlas that you are developing in eBrains. That is really according also to the audience that I have to say that it has almost doubled in the last 10 minutes. It means that you are proposing something very attractive for our audience. So thank you very much, and the floor is yours. Thank you.
Thanks a lot for the introduction. I must say that was the first introduction in Spanish. I enjoyed it a lot. And thanks a lot also for the invitation. I feel very honored to talk here in this audience. Please interrupt me if something is going too fast or if you have questions. That is not a problem. And I think the most important things about myself have been said, although I didn't understand the details. So I will share my screen and start right away. Do you see my screen now?
Yes, we can.
Yes.
Okay. So I will give you an introduction to the mostly to the human brain atlas in eBrains, but I will start with the general setting of eBrains brain atlases as such very quickly. As was probably introduced, I'm heading the service category 2 in eBrains, which means the eBrains Atlas services, which comprises I think 4 tasks in the work package 4, if you are familiar with the work plan in HBP. So the eBrains brain atlases, you find the most information about it now finally on the eBrains portal. So if you go to eBrains.eu, you quickly find there under services the Atlas service category, but there's also a direct link as you see here, and we have described the basics now on the portal page. So this is the place to start, and as you can see there, we are developing currently atlases of the mouse, the rat, and the human brain. And there's also an atlas in development of the non-human primate brain, which is not yet released. We have just started this project now in SJ3, but at the end of SJ3, there will also be a basic and extendable monkey atlas. Here on the eBrains portal page, if you would scroll down, you can already see here that the eBrains Atlas services not only contain the atlases as such, the human and the rat and the mouse atlas, but a range of tools to work with these atlases. And we will look at some of these tools today. But just briefly, we have 3 categories of tools. The one category are tools to really interact with the atlases as such, to access the atlases and work with them. Then there is a second category of tools that allow to integrate data with atlases. And the third category are analysis tools that allow to perform certain analysis with the help of brain atlases. In my talk, I will focus on the human brain atlas, and I will focus on the tools, on the interfaces to work directly with the atlas. I will not talk so much about the data integration tools and the analysis tools, although I will show a few of them. But before we start, it's usually good to have a common understanding of what an atlas is, because that is not, sometimes people have different perspectives on what an atlas should be. And I always like to show this in conjunction with a atlas of the Earth. So when we talk about a brain atlas, we typically assume that there is a reference space, a coordinate space in which an atlas is defined, just as we have in an atlas of the Earth, we have the Earth as a coordinate system. And then we have maps of regions that basically give a label to different locations in this space. Just as in a map of the Earth, you might have labels, for example, of the different countries, of course. And third, we assume that there is a taxonomy that gives names to these different regions, and that possibly describes relationships between them. In the brain, this is very often a hierarchy of brain regions. So this is kind of like a minimal definition of what an atlas is. And there's one important aspect also for atlases of the Earth, that there's not a single map. So if you have an old book, Atlas of the Earth, you find, of course, different maps that might describe the climate or the topography or the politics. When you flip the pages, you can compare how this relates between the different maps. And especially for the human atlas, we have this ambition of building a multi-level atlas that provides different such maps and even more. So this is important to understand. We do not consider an atlas as a single map. We consider this as a map or as a parcellation. When we talk about an atlas, we assume that there might be multiple maps defined in the same framework. And I will try to get to this now. So for the human atlas, we often say the multi-level human atlas, our ambition is that this framework captures the many facets of human brain organization, not a single facet. The many facets of the human brain organization in one framework. And this includes three levels, three primary levels. One level is we want to link different spatial scales. In particular, we want to build a link from the cellular scale up to the whole brain scale that is typically used in neuroimaging or clinical applications. And second, we want to provide multiple maps of the brain, which are complementary to each other. I will get to this in more detail soon. And then there's a third important aspect of this paradigm. We want to link multimodal features to all these brain regions definitions. So the atlas is not designed to just give an orientation where certain regions are in the different maps, but we use it as a framework to link and aggregate information about each of the brain regions in the form of multimodal data features. So really in the form of neuroscience data sets that are linked to these brain regions so that they describe each brain region in different aspects. And I will get now to these three different points in sequence, starting with the aspect of multiple scales. So how do we link these spatial scales in human atlas? How do we build a bridge from the single cell level to the whole brain level? And the way we are currently doing this is that we start from post-mortem histological data sets where we have stained histological sections that show us the distribution of neural cells as you see it here. This is at a resolution of 1 micrometer. So we have many data sets and studies starting at this scale. And if we zoom out to a level, let's say at 20 micrometer resolution, it looks a little bit like this. We see that there are cortical layers in the brain that are characterized by the architecture or by the arrangement of cells. You see here parts from two different areas on the right, from two different brain areas. In fact, this is the primary and secondary visual area. And you can see how the pattern of the cells here in the cortex is different because they arrange into layers. And the way that these layers are structured and show presence of different types and sizes of cells, we can distinguish such areas in a structural way. And this gives us an idea also about differences in the functional characterization of these areas. So we can, at this scale, characterize different regions in the brain. And when we zoom out further to the whole brain level, this is still the same picture that we see here. We have worked to produce a 3D model of a complete human brain that comprises data at this scale. This is a so-called big brain that has been developed by our lab together with the lab of Alan Evans in Montreal in Canada some years ago. So this is currently the only 3D model or 3D image of a complete human brain at microscopic resolution. It has been reconstructed from more than 7,000 histological sections into 3D and is now freely available as part of the eBrain's Brain Atlas. So when we look at this, this is the Atlas viewer that we have developed, which is online, so you can easily access this yourself. You see here that we can rotate and navigate in this 3D brain model. It's a data set of about 1 terabyte size, but you can just through an internet connection, it can be explored in 3D. And as you have just seen, we can also rotate it. So it can on the fly compute oblique sections at any angle in this brain model. The important point is now here, this model, since it is available as a full 3D human brain, we can relate it to MRI scale template spaces, coordinate spaces, but we can still zoom in to the level of single cells. But this still doesn't really give us a link across the scales. The very important aspect is that we have these cytoarchitectonic regions that we have just shown you a border between two such regions. And what we are doing in this brain model here is we produce maps at the full microscopic resolution, maps of cortical layers. This is also done in cooperation with colleagues in Montreal and in UK. So there is a mapping of all the 6 cortical layers across the whole brain available. And further than that, we are also mapping the borders between these cytoarchitectonic regions that we saw just a while ago in this same brain model. This is what you see here. So to map such a region, this is the primary visual cortex here, the one you see in almost pink. This requires to find the boundaries of these regions in this case in about 1,500 histological sections. And we have done this by learning with AI methods, learning from neuroanatomists to do this, because it would not be feasible for a person. But we are doing this currently, and we are adding area by area to this microscopic brain model. So we know exactly where the different cytoarchitectonic areas are in this microscopic model. And now the point is, if we go from here to the classical scale of 1 millimeter that is used in neuroimaging and so on, and where we have multiple scans of brains, we have the problem that, of course, this primary visual cortex in the big brain is only the map of a single brain. And if we would map these same areas in different brains, you see here 10 different postmodern brains, which have not been fully mapped, but only in a subset of the sections, you see that the areas have very different shape and location and size. So the morphology of these areas in the human brain has a strong variability. And we can, of course, not just use one map to describe how the brain is structured. So what we do in Jülich is we develop the so-called Jülich brain probabilistic cytoarchitectonic atlas, which contains maps of many postmortem brains, typically 10 for each region. And what we then do is from these different individual maps, we build a probabilistic map by averaging it in a standard space, which is defined at the millimeter scale. So we get a probability distribution of where this area most likely is in a standard space, and what the probabilistic distribution is. As you see it here this is an example of the frontal pole. And from this, we can again build a map if we take the maximum probabilities. Now, the important point is that these definitions of the same regions in the brain, they give us a link across the scales. They connect the microscopic scale to the neuroimaging or even the clinical application world. Since we have these probabilistic maps that capture some aspects of human brain variability at the millimeter scale, and which are available in the standard space, in the MNI standard space. But we have, for the same definitions of the brain structure, we have also in this individual brain very, very detailed maps at the microscopic resolution. So we can build a direct link. We can, from this big brain model at the microscopic resolution, we have the possibility to relate it to the millimeter scale and to the population level, because we have the exact same definitions of the structural regions in both spaces. And this is one way for us how we connect the different scales in the atlas, by putting such different spaces and region definitions into a common framework. Then I said the second level is that we want to provide multiple complementary maps. And the atlas, as it is currently online, already supports quite a range. So here you see also a screenshot from the atlas viewer, and it shows the cytoarchitectonic maps at the millimeter scale. And there is a browser where you can then, just as you have it in Google Maps, where you can select different layers of this map of the brain, and also different coordinate spaces. And it's a bit small probably, but you see here, there are also maps of functional structure, functional activations, and maps of fiber bundles that can be selected. And here we see a map of major fiber bundles in the brain coming from our colleagues in France, Jean-François Magin. And here's a map of functional modes. Both of them have been computed across large neuroimaging cohorts. So they capture the variation across hundreds of individuals in terms of function and in terms of fiber architecture in the brain. And they're integrated in the same atlas. And of course, you have this then also coupled with the microscopic big brain model, where we also have these cytoarchitectonic maps defined. And this is all accessible in one framework. And if you want to go to the high resolution, you can do this by choosing the big brain template and zooming in. But it's all connected with each other. The third level, the third aspect of this multi-level atlas framework, as I said, is to link many different neuroscience data sets to specific brain regions. And I will get to this now. So the idea is that we have many labs, also external researchers, sharing their neuroscience data with eBrains. You have probably seen presentations about the eBrains knowledge graph and the eBrains data sharing services. So we are relying on many data sets coming in through this curation and data sharing system. And in the data curation, we have an explicit process that does the best attempt to assign location to the data sets that are coming in. And if the location can be clearly assigned to brain regions according to the atlas, then we can consider such data sets as features of the brain regions, and we expose them explicitly through the atlas as multimodal characterizations of the brain regions. And here's a little movie that shows how this works in the atlas viewer. I get to this a bit more. So you can select here brain region, and then there is here on the left, sorry, I just run this again. Once a brain region is selected, you get a context menu, and you see there regional features, and it lists us features. For example, here we have linked intracranial EEG recordings in the standard space, where we then find the recording sites that cross this particular region. There are more examples. For example, we have measurements of neurotransmitter densities from histology, which are also linked to these brain regions. You see here how we can access receptor transmitter distributions for different neurotransmitter receptors for this particular brain region that has been selected. And it's just there at your fingertips, although there are years of lab work behind all of this. And here you see how functional fMRI scans, functional activations are listed, which have a strong overlap with this brain region. And lastly, you see here brain connectivity. So the connections from a particular regions to other regions. All of these are features that characterize these brain regions that are based on neuroscience data that has been shared in eBrains, and it has been carefully linked to the atlas and is now available at a fingertip to describe the brain regions in their different facets. And of course, these same links are accessible from the microscopic scale, given that we have the same regions defined at the microscopic scale. Just to repeat myself, we are currently increasing piece by piece the coverage of maps that we have accessible at the microscopic resolution. And all these data sets, these characterizations of the different brain regions, they are of course available for download to users. So you can download them through the data sharing system of eBrains, but also via the atlases, you get a direct link to the same resources. So there's actually even a kind of a shopping cart implemented in the atlas where you can, while you navigate the brain, you can just click on data sets that are of interest and later on just download the whole package of data sets with proper documentation and metadata. So this is in short form the principle of the multi-level atlas, and I just would like to show you how this is embedded in the infrastructure that eBrains provides us. I have already briefly touched this point. So everything, every content of the atlas is stored in the eBrains knowledge graph. This means everything you saw, the maps, the template spaces, the multimodal features, are themselves data sets that have undergone curation by the eBrains curation services that have rich metadata attached to them, and they are listed as data sets publicly available in the knowledge graph. And this goes really through the hands of real people, these curation support teams that we have in eBrains. And you can find all of these data sets by searching on the eBrains knowledge graph. So I think you have seen that here before, the knowledge graph. If you like, I can also show it quickly. You might just raise hands if you're interested, then I will do that. So these are really, each aspect of the atlas is a fair data set, so to say, which you can find with citation policies and metadata on the knowledge graph. The important point is that the atlases now just point back to this. So the atlases are, you can be considered as a framework that connects data sets from the knowledge graph into an anatomical context, into an anatomical framework, makes them accessible there. So it's not separate. It is an anatomical framework around the open data sharing that we are doing. And these atlas services, I will come to some of these software tools soon. These atlas services, they have also access, they make also use of the supercomputing systems that eBrains provides. So all these, in particular, all this analysis of the microscopic scale data is performed on supercomputers typically in the through the eBrains infrastructure. And typically when you access it from the atlas, you already have some form of derived data that is a bit smaller and easier to handle, but not only. The atlas is also designed to give you access to big data, which is a bit more involved and which is a focus now in SGA3 in the current project phase. And what you mostly see from the atlas, what you have also seen now in my slides, are two interfaces to access this atlas. One is an online viewer, an interactive viewer. We will soon give it a new name. It will be called the Brainscapes Brain Explorer, Atlas Explorer. So this is what you have seen on my slides. Everything is accessible through this online Explorer. But there are also other ways of accessing the atlas. And one is very important, and we are currently building this since half a year. We are on it now. Is a Python library that allows researchers to access exactly the same functionality, but without interactive work in a web browser, but instead in a programmatic way. So you can here through Python, so through programming language, you can access the exact same functions. You can instantiate the atlas, you can select different maps and parcellations, you can query data features. All these things can be done fully programmatically and reproducibly without using the viewer. Yeah. I typically show the viewer in such a presentation because it makes things obvious, but the viewer is only one way to interact with the atlas. There is a programmatic way. And of course, there's also, if this means something to you, there's also a REST API, so an HTTP interface that can be accessed by other external applications as a third way of accessing the atlas. So this is a brief overview what in particular the human atlas is. Maybe before I would like to get to a few use cases. I think I have still some minutes left to just explain to you how what we see as some important use cases of the atlas, and also what currently drives the design of our developments. We especially since the last year and in the remaining 2 years now of Human Brain Project, we consider it as extremely important that we now have clearly user communities in mind for finalizing these software interfaces and these developments. And here are some, not all, of the use cases that we have in focus. One is experimental neuroscience. So in particular, since we support this high resolution reference space, this Human Brain Atlas in eBrains provides a way of integrating data from histological experiments. So many labs, they produce measurements of parts of the brain, partial, like a piece of the cortex, a piece of the hippocampus, whatever, and study it with a certain imaging modality, but it's very difficult to make use out of it in conjunction with other data sets because they are not in the same space and not in the same system. And we consider this atlas very important to integrate such high resolution data into a common anatomical framework. I will show you an image very soon about it. The second group is data analysts, like we have many in the neuro imaging community, for example. So these atlases are an important resource to run reproducible experiments that require assessing data in anatomical context in relationship to certain brain regions. And we see the atlas as a very important resource there because it has a very comprehensive capture coverage of different aspects of brain-oriented organization, which goes beyond many of the currently available atlases. And one community that I find very important and exciting at the moment, and it's actually two communities that share some commonalities, is brain simulation and also artificial intelligence. In the AI field, we have currently a large sub-community, if you will, that works on biologically inspired artificial intelligence methods. And just like people from brain simulations, they are interested to understand the microscopic structure of the biological networks in the brain. So they're very interested in knowing what are exactly the densities of cells in the different layers of certain brain regions. What is the connectivity inside a brain region? What is the number of connections coming into a certain brain region? Things like this are very interesting and important for setting up brain simulations and for designing biologically inspired artificial intelligence systems. And these atlases that we have can provide this data. So we are in touch with these with these research communities, they're very interested in this information. Of course, another important group that we are targeting are hospitals. The atlas can be used to plan surgeries, of course, also to compare disease, compare disease to healthy brains, to localize findings, and to assign anatomical location to findings. There's actually a clinical toolbox in development currently that would allow, for example, to easily integrate a scan from a patient, an MRI scan from a patient with that describes certain parameters, certain measurements, to align it with the atlas, possibly even without uploading it into the system, and then to superimpose it with all the information in the atlas that I have shown to you. All the maps, the microscopic resolution features, the functional features, and so on. So there is a clinical toolbox in development that we are planning to release next year. And of course, an important group is also education. So you can use these atlases nicely for studying brain anatomy and other things. Let me just come to this first point of data integration. So here are typical examples where labs measure certain regions of the brain with different modalities, and what you typically get is something like this here. This is a result from a histological experiment, and if you look at it like this, it is very difficult to make meaning out of it because you don't know the context, you don't know the location in the brain, you don't know the orientation, anatomical orientation, and this is a piece of the hippocampus. And we have tools in the atlas that allow to integrate it with the reference model. Yeah? So in that case, we have superimposed it at the correct location in the big brain model. This is not a pixel exact superimposition. This would allow to completely unfold the hippocampus and fold it back into the big brain model. But it is an integration at the level where the major orientation is correct, the position is correct, the hemisphere is correct, and so on. And so you see already that this makes this data set much more useful. Yeah? It's now better interpretable, it's better findable, it's better usable. And this is exactly what we are envisioning with the atlas, that we can better bring multimodal, heterogeneous results from neuroscientific studies into a common framework to make joint use out of that. And this, in particular, this is a tool called Voluba that you also find on the eBrains webpage. I'm not going into detail on this. And this is probably my last slide. Data analysis. Here's one example how we use the atlas to perform data analysis. This is the so-called JUGEX tool. This is a workflow that allows to perform differential analysis of gene expressions in different brain regions. And this, we have implemented this in the atlas in two different forms. One form is a plugin of this interactive atlas viewer, where you can really interactively just load the web page, select brain regions by clicking them, enter some candidate genes that you're interested in. And what the system will do is it will retrieve these gene expression measurements from the Allen Brain Institute, filter them with these atlas regions, and perform a differential analysis to tell you whether these genes have significant different expressions in the two brain regions. And by implementing this into the atlas, it is also available for programmers through the Python client without having two different implementations. So this just comes by design, that you can use this in a programmatic workflow, and you can also use it online in a viewer. If you are interested and we have more time, I'm happy to show it live. Currently, I just step over it. And then I think this is actually the last slide. These are examples of data where this brain simulation and AI people are very interested in. So you see here polarized light imaging data, which exposes at the microscopic level fiber, the orientation of nerve fibers. Here you see, as you saw in the beginning, distributions of cells. You see densities of cells, sizes of cells. Here you see distributions of neurotransmitter receptors. And here you have a staining that distinguishes different types of neuronal cells. This is the kind of data that we are sharing and providing to these communities through the atlas. So they can select brain regions and access such data features to study the characteristics of different brain areas. So to conclude, I give you a high-level picture. This is a very young slide and maybe a bit high level, but I thought I'd share this with you. So we are here today. We have now an atlas that is openly accessible. It has many maps that stem from lots of delineations in histological experiments. We have probabilistic maps from hundreds and thousands of in vivo imaging data, and we have already quite an impressive range of multimodal data features linked to regions. Until the end of the official HBP, our main ambition is to massively increase the coverage with such data features, and also to implement tools for some specific use cases. I mentioned to you this clinical use case that we want to support better until 2023. And beyond, this is a bit what we are dreaming or aiming at. It of course requires us to get the funding and to continued support. We want this to become a community-driven reference framework at the single cell resolution. That means we want to go to an even higher resolution with this reference model, and we want to open it for more extensive contributions from the international community in terms of integrating that data with the system, integrating different maps of atlases with the support of the curation teams that we have. So I'm really hoping that we can keep this up, that we have experts for data curation and data integration that can support the neuroscience community in integrating the data in such a framework here. Yeah, by this I'm at the end of my talk. I hope I was roughly in time, and I'm happy to answer questions, or if you like also to show you some things in a web browser live, whatever you like. Thanks a lot.
Thank you, Timo. Also related with this further questions, you can probably share, or later we will share your email in case that somebody wants to address you something very particular.
Of course.
Thank you for really interesting presentation. It is really a showcase window of what the HBP is doing, because here is somehow connecting all the works that the other HBP groups are doing in terms of providing data and so on. Is fantastic. Well, I leave the room for people that wants to ask any questions to Timo. So please, if you have any questions.
I have one question for Timo.
Thank you.
Sorry, let me turn on my camera. Yeah. Hi, so this was great. I really loved this. Looks like a very useful tool that I can only hope it's full and out very soon. So I'm particularly interested by this Python API that you were mentioning. But in general, so my question is how complete is this at this point? So I just, as you were speaking, I just got in and I started looking and I couldn't find my area, the area I would be interested in, which would be area 46. I don't see it there. Is it because this is just growing? Is it something that we're going to see happening? Or that part is complete, and what we're after now is extending the tools around. So how is it complete, or is just that something that is on the process of being built at this point?
So we are in a process, and this will never stop to be a process, of course. But the cytoarchitectonic atlas is now complete in a sense that we have a full coverage. You see here the cytoarchitectonic maps as a maximum probability map here on the viewer. It's currently complete in that we have coverage of the whole complete cortex, but we have still some what we call gap maps. You see this one here in the frontal pole, which we are still working on with neuroanatomists to find the exact subdivisions of this and map the exact subdivisions. So you may not find every area, but this should be the most detailed one that you will currently find, and it is always increasing. So I'm not a neuroanatomist. I cannot myself easily respond to your remark about area 46. But what so when you look here, you see the full hierarchy of the system, and you can search through it. And I guess that 46 you would have found it if you just search for 46. So one problem that we have when using Atlas is that people use different terminologies very often. So it might be that your area is there, but with a different name. It might be that the Atlas is more fine-grained than what you are looking for. So you find the subdivisions. And this is something we are constantly working on. Not myself, of course, but in this case, it's a group of Katrin Ammunds in Jülich, to also make clarify these different namings. And this is a very complicated issue because sometimes people just use a different name for the same thing, but more often people use the same name, but they don't mean the same thing because they refer to the same area mapped under different constraints or after different principles. And this is something that we are very interested in, and we are hoping with the Atlas to actually reduce such problems. Yeah? But jumping into this whole problem domain also means we make these problems also explicit, and you hit them. Yeah. So long story short, if you didn't find area 46 here, I would be extremely interested to take this request. I mean, I have taken a note already, but there is always this support address, and we hope to be in touch with users like you who would then say, we're looking for area 46, didn't find it, and we would be very happy through the support system to respond to this, yeah, and clarify this. Yeah, we also try to work on homologies between species, which is very tricky also because of these different naming schemes. Yeah, but you were also asking.
There are different terms, so I will scan. Now I see how you got this list. So probably in the list I can find maybe one of the other terms that has been used for this area, right. And I can.
So typically, like if I say 44, you see here already. You see it's area 44. It's IFG, inferior frontal gyrus. So there is a certain level of this already, but 46 is maybe really just now. Even 45 is there. Yeah. So what you could do is, if I assume that the 46 is close, you could now just look what else is in the inferior frontal gyrus. Yeah. So we could here.
You don't have to do it now.
Yeah, this is the way you would approach it. Yeah.
Yeah. Very interesting.
Yeah. And maybe
if I may ask another question. So about the Python.
Yeah.
About the Python. Tool here, so I was able to install it and start looking at it. So it's available. You say you're working on it, but I see it's up.
Yes, it's
available. I can use it, or I should think it's still buggy and not wait.
Yeah.
What is your recommendation?
I'm working after the paradigm to expose things early. And honestly, so this is very young. It is not stable at all, and there might be errors. And you should, when you look at this, let me just show it to you. So when you go there, it will. This is actually here. What you found. It tells you that this is still at the prototype stage. The API is not stable. So you're welcome to play with it and test it, but you have to be careful because in the course of this year we will still make heavy changes. Yeah, it works, but it is not deeply tested yet, and we might at any time break the API and change things. But I'm happy if you try things out and if you're in touch with us, and because I'm convinced that being in touch with users early helps us to make it better.
But say the maps that it retrieves, they are correct. What might break at some point is that the code that I generate stops working because you change something.
Yeah.
But not that my results will be buggy because the map is not the one that works.
I think it's not very buggy, but this is a prototype, so we don't take any.
Okay. Yeah, I see.
Yeah, it's.
I'll play with it.
we didn't test it as much as other things yet. Yeah, but it delivers the same data sets that you will find in a knowledge graph. So once you have a data set, this is a curated data set, of course. Yeah,
it's the same as you would get in.
Yes. 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 Exactly.
Excellent. Thank you, Timo. Very, very, very exciting. Very nice.
Thank you.
Any other questions? Well, Timo, I have a question in that case that you have mentioned the three main points that are structuring this excellent tool that is the multiple scale, the multiple maps and multifunctional features. What is the one that you think this makes the difference between this atlas and the other atlas alternatives that you will find worldwide? What is if you have to choose, what is the main potential, the main functionality that distinguish this atlas from other options?
I think the biggest strength is the integration of these different multimodal data sets, because this is really something that will be growing and that you don't find so easily elsewhere in the same structured form. Then comes the link across the scales. I think that is not available anywhere else. Yeah? You have some of these aspects covered, for example, by the Allen Brain Atlas. They have these gene expressions linked with histological sections, and they do that very well. But there's a clear focus on that part. But HPP is here going very broad with different modalities. So I think this is the strongest part, but closely followed by this coverage of microscopy at full 3D. And these complementary maps that I've shown, the third part, this is certainly something that is not so difficult to implement. Yeah? So this, I would put that on as last in the list.
Okay, thanks.
Timo, I have a question on the possible segment of users you are thinking on. You said initially that there were several use cases that could be used. One is on education, on neurology. But do you feel that this kind of tool could be also part of a wider structure for companies in order to test in research related to pharmacology? Or do you feel that it's not easy for large, big pharma companies to be able to use it as a mechanism to improve or to shorten the time necessary to create a new drug?
Is it a very good question. So first of all, we have a few more use cases in our list. I didn't present all of them, but the pharma case that you are touching here is a very interesting one. So I must admit that I'm not very experienced with pharma applications. And I had some interactions also. That there was a while ago, there was actually a workshop, community building workshop, where we met some people from pharma. And my impression is we need to discuss this more, because typically they ask where are the disease cases here in your system. And on our side in the research, we are addressing to understand the healthy brain. So first of all, what you see here mostly describes the healthy brain in its different facets. And I think it is very interesting because you can link the diseased cases to the atlas and compare things. But it is difficult for me to understand how this use case would really look for typical company application in the pharma industry. So it's difficult for me. My impression is that the larger pharma companies, they have large data data sets themselves, which cover exactly the diseases and the problems that they are interested in very well. And so the bridge is not obvious to do here, probably. Yeah. I could imagine that there is a big potential, but I honestly admit that I would need more, maybe face-to-face discussion to carve this really out.
Okay, because one of the things we can do is just to organize meeting with a few people from the pharma industry just to discuss with you about the possibilities for the future. Because at the end, my feeling is that they could also interact with your next roadmap of activities, looking for some kind of interest and use cases coming from the big players in big pharma.
Yeah. Very open to this, of course.
Okay, thank you. Bueno, estamos ya a 2 minutos. Just another question. Is there anything else?
If I may, one last question. So I was wondering now when you connect from the macro scale to the macro scale to the micro scale to cellular histology or physiology data. I presume that data is mostly from animal studies. So how do you link the two? Is the link, they're not direct correspondences between areas, or it's harder to do that map. So how does that, how do you map those across species to make the jump between areas?
Yeah. So currently we do everything in human, in fact. So all the high resolution histology things you see here is all done in the human. But of course, there are certain things that you cannot do in human. The most prominent example in my work is always tracer studies that show you exactly connections in the brain. We don't easily get this in humans. And for these parts, it's difficult. Let's put it that way. What we are building is a catalog of homologies. That is right this year going on. Just build a catalog of cross-species homologies that are well understood or at least disputable. And this catalog, my idea is to actually integrate this catalog in the system, so that the system could tell you, well, for this area, besides all the features you see here, there are also some known homologies to other to animals, and click here to just click here to see what you find there. On that level, I will integrate it. But then there's a second level to this story that we are not doing inside the atlas. So in the research, people often try to build statistical models between certain data modalities that you have, for example, in the mouse or in the rat. And they see if they can apply these models, these machine learning models, for example, in the human, where you might only have one of these modalities. Yeah? It is very difficult to integrate something like this in the atlas because it is not clear whether this is correct usually. So this is typically something that I have some projects ongoing where we do such things, but I would be hesitating to make this a part of the atlas because it's, until now, the atlas is really, it has, without exception, it's all real measurements that you find here. And we honestly describe where we did these measurements. But if you bring in such a machine learning model, it's not anymore measurement. And it would be a matter of discussion whether you accept that this is correct or not. Yeah. So I think there is a level on top of these atlases, which is filling gaps in the data with the help of some generative predictive models that I wouldn't see directly part of the atlas.
Okay.
If this answers your question.
Yeah. Very interesting. Thanks.
Timo, just to say the rest of the faces, do you need again your screen or not?
No,
no, if you need it, no problem. So just to see more faces here.
I didn't even realize it. Yeah.
Yeah. But just to 2 p.m. clock, then, well, first of all, on behalf of the UPM, thank you very much for participating here this time. We had, and they speak more than 54 people. It was a very good and huge number. Then congratulations, you have attracted the interest to many persons here. Then, no sé si hay alguna pregunta más. ¿No? Bueno, pues entonces vamos a cerrar aquí. Muchas gracias a todos por vuestra participación. Thank you very much, Timo, for coming virtually. I hope the next time we'll have the opportunity to see you face to face. Bueno, gracias a todos.